and hear me well? I'm taking it as yes.
Yes,
so,
yes.
Thanks for
Thank you.
Yes.
Yeah, thanks for kind invitation. It's fun to be at STC scientific workshop. And in today's talk, I'll be discussing about our work, which is linked to development and cancer, and how we are using spatial transcriptomics and its clinical implications. So our lab is interested in understanding oncofetal ecosystem, and as the name suggests, oncofetal means something which is shared between a fetal tissue and tumor, and ecosystem is a geographical area. So we are looking at similarity between fetal and tumor tissue from microenvironmental perspective. And when we are looking into human development and cancer, so cancer occurs at mainly two stages. Either it occurs at childhood, these are pediatric tumors, and they have shared some similarity with the neonatal development. And most of the time in these cases, there's a maturation block, which means that developmental genes, which should switch off at some point of time, they do not switch off, which lead to the excessive organ growth. For example, as Wilms tumor 1, where WT1 should turn off at some point of time, but it does not. Now, when you start comparing adult tumor with a fetal tissue, and this is what we did in our publication a couple of years back, we found that there are some similarities between fetal tissue and tumor. Also, the adult tumors are very different from pediatric tumor because they have seen so much wear and tear of the life, and they are mainly caused by successive accumulation of the mutation, which could be further fastened by exposure in the life, such as viral exposure in case of liver cancer. And this is something which is called, we call oncofetal reprogramming. Now, the concept of the oncofetal reprogramming is almost 6 decades old, and the first time it was observed in HCC, where a protein called alpha-fetoprotein, as the name suggests, the protein is present in the developing fetus, and it's one of the markers present during pregnancy in the urine of women, right? And it's secreted by fetal tissue. But in a lot of the HCC patient or liver cancer patient, there is a reemergence of this AFP protein in the tissue, suggesting that there are some fetal-like programs reappear in the cancer. And further, this was shown in many other tumors, such as CA125, which is a biomarker in the breast cancer. But all these studies were looking at the tumor cells. And as with the advancement on the single cell genomics, we are now have much better tools to understand various components of the tumor, including its microenvironment, immune cells, and stromal cells. So our study basically tries to look into the fetal-like properties of the non-tumor cells or the microenvironment, how the stromal cells become more fetal-like, or how immune cells become more fetal-like. And we are trying to understand different components of the oncofetal ecosystem. We are also trying to understand the cell-cell interaction and signaling pathways between them, and how it may lead to the immunosuppression, which may have implication for cancer immunotherapy. And finally, if we can use oncofetal ecosystem as a biomarker for cancer immunotherapy. So this journey started a couple of years back, where we first time using comparative spatial transcriptomics by looking into tumor tissue, normal tissue, and fetal liver tissue. And we used some mouse models to do some validation. And what we did in this study is that we are looking at cell types which are shared between fetal tissue and tumor, but are absent in homeostasis. So essentially something which is not part of a normal liver was present in the fetal tissue, but reappear in the cancer, and which transcription factors plays a role in this, and how does the ecosystem look like in this case, right? So for this, what we did, we looked into the human liver cancer, HCC in this case, divided this into each tissue into different segments. We call it T1, T5. So looking at the spatial heterogeneity by doing single cell RNA-seq. We also look into the adjacent normal tissue to see how tumor is different from a almost healthy adjacent tissue, and how these cells are similar to a growing fetal liver. So we look into the second trimester of the human fetal liver, and we look into cells which are present in human fetal liver at 14 week, 16 week, 18 week, and 20 week of the development. And essentially what we found is that we could identify different subpopulation in the human liver. and we could also find how these cells are different in normal versus tumor, how these cells are different between the core versus periphery, which is basically a spatial information of the tumor. As we know, the core of the tumor is much more hypoxic compared to the periphery, which is more vascular. So we identified different population in the core periphery as well as normal versus tumor. But I think this was the most interesting bit, where we could also identify a population of macrophage we call TAM1, tumor-associated macrophage 1, which is shared between fetal liver tissue. So we find the very nice correlation between the gene expression pattern of fetal liver macrophage and a population of tumor macrophage. So I'm not going into the detail of this study because it's published, but there were 3 different populations of the macrophage, but only one of the macrophage population was similar to the fetal liver. Similarly, when we look into the endothelial cells, also we identify among the different endothelial cells present in the human fetal liver, we could identify endothelial cell, which is PLBAP positive, is also shared between fetal tissue and tumor. And in those days, we did a very targeted spatial transcriptomics approach, where we're looking at 96 genes, which were present in the tumor immune microenvironment, and some of the genes which are present in this oncofetal ecosystem. And we could find that there are genes which are shared between fetal liver and tumor tissue, and we call it oncofetal high ecosystem. And this has similarity with a cold tumor features, such as they have high fallout-2 positive macrophages, PLBAP positive endothelial cell, high NOT signaling, VEGF signaling, and FOXP3 and CTLA4 positive T cells. On the other hand, when we look into adjacent normal tissue, we do not find some of these oncofetal features of the tissue. Now, this is the model we presented in 2020, where we showed that how a tumor, which is in homeostasis, does not have these PLBAP positive endothelial cells and fallout-2 positive macrophages, goes back to fetal liver developmental stage, where it now starts appearing with more fallout-2 positive macrophages and PLBAP positive endothelial cells. And since this work is published, I'm not going into the detail, but our mechanistic model was that hepatocytes are a very interesting population. They do not divide in homeostasis, right? So they divide during fetal development, which is the organogenesis process, or they divide during injury, or they divide in cancer, right? A dividing hepatocytes make VEGF, which lead to the endothelial reprogramming and bring these PLBAP positive endothelial cells, which now reprogram monocyte, which are coming into the tumor, into a more embryonic-like state, which is fallout-2 positive macrophages. And all these things were also shown by in vitro experiment in the paper. And then these macrophages are interacting with T cells, leading to immunosuppressive tumor microenvironment. And when we were concluding this study, there was a very interesting clinical trial, which was approved as the FDA drug now, okay, which is Exo plus Peva. So this is an anti-PD-L1 and anti-VEGF inhibitor for liver cancer patient. And as you know, liver cancer is one of the most aggressive cancer. 80% patient will die within 5 years, 50% patient relapse within 2 years. So it's a very aggressive tumor. And there is one of the reasons it's aggressive is because there is no systematic therapy. So in 2020, there is the first systematic therapy, immunotherapy, which was approved by FDA, which is anti-PD-L1 and VEGF alpha. And as I just mentioned in our model, that both PD-L1 and VEGF alpha are component of oncofetal ecosystem. And that's where we started thinking that oncofetal reprogramming might make tumor more immunosuppressive, but will also could serve as a biomarker for immunotherapy response. So since then, we are looking into how these oncofetal cells could play an important role in therapy response in HCC. And HCC comes in 3 different stages: early stage, intermediate, and advanced. So early stage is something where there's a best chance of survival, and it could be cured by surgery. But what happens is that 50% of the patient will relapse within 2 years, right? Some patients will relapse as soon as 6 months or 9 months. And when these patients relapse, they are given either a Lenovo or an Exo plus Peva therapy. But again, there are some responder and non-responder. So this is what we are trying to understand. Can we use oncofetal ecosystem as a biomarker to understand which patient will relapse, will they respond to therapy or not? And this is the part which I'm going to present in next 10 minutes or so. And this work is currently under review. So what we have done, we have used our previous atlas to understand different populations. And we have came up with an approach where we could now, if we did this experiment on 14 patients, but to understand the clinical feature, we needed a big cohort. So went back to this cohort, which I'm working with my Singaporean collaborators at NCCS in Singapore, where we have just developed this big cohort of 147 liver cancer patients. And for this particular experiment, we have used 68 patients from this cohort. This is a mature cohort. We know what subtype of this liver cancer in these patients is. Are they viral positive, viral negative? What happened to these patients after curative surgery? So this is one of the very unique HCC cohort in the world, where most of the surgeries were done by a single surgeon, PS job. So we did this, we came up with cell type specific expression, and we used this approach of deconvoluting bulk RNA-seq data. And we came up with something where we know that which patient has what kind of cell types present in them, right? So by doing this, we could divide this data set into two different groups by looking at patient who relapse versus patient who does not relapse. And we could identify the proportion of various cell population. And we found this group very interesting because most of the patients in this group relapsed and they have very high Oncofetal score. And I'm not going into the detail how we calculate the Oncofetal score because this work is still in review, but we could identify cell types which are associated with recurrence in these patients, right? So next, seeing is believing, and that's where I think this spatiotemporal consortia will be very powerful. So what we did, we were the two patients where we knew the outcome. So one of the patients where there was no relapse even after 3 years, another patient where there was a relapse within 6 months. We went back to these two tumors and we did selective spatial transcriptomics by looking at specific areas in the tumor. And we were guided by markers, tumor, immune, and stromal markers, right? And we did the same experiment for human fetal liver. And to cut the long story short, what we find is that a patient who had relapse within 6 months had more areas where we could find more of these oncofetal cells compared to a patient where there was no relapse. And this is where we started talking to BC in BGIS Australia, where we developed this collaboration of looking at this oncofetal ecosystem at a single cell resolution by this powerful technology STOmics, right? So what we did here, we use two human liver cancer tissue, HCC, again in this case, and this is the data of like number of genes and all we are detecting here. And we could go to very small resolution to understand individual cells, which is not possible by other technology as of now, right? So we could identify different tumor and non-tumor cells in this cluster, but also we could look into some of the interesting genes which we are interested in, such as macrophage marker, CD68, CSF1R, endothelial cell marker, PKM1, fibroblast marker, ACTA2, and CD4 and FOXP3, etc. But we were more interested in genes which we think are more associated with fetal-like reprogramming, and these are the genes we are looking at in these two tumor tissue these are 3 different type of genes which are associated with 3 different fetal-like cells. And we could find that these genes are present in this HCC tissue. And this is basically how we identify these oncofetal cells in this STOmics data. But next what we did this neighborhood analysis, and we are now trying to come up with a neighborhood of oncofetal neighborhood. So we want to understand where these multiple oncofetal-like cells co-localize with each other. And that's how we identify oncofetal niche. We say, if you have 3 different cell types which are fetal-like, are they in close proximity? And in that case, we'll call it oncofetal niche, right? So we could identify this oncofetal niche in both HCC tissues, which we sequence here with BGI, and we could come up with this neighborhood index. Now, in this oncofetal neighborhood, we could also identify different oncofetal cells, oncofetal immune cells, stromal cells 1 and 2, and we could call it oncofetal high versus low area. But since we knew that which patient relapsed versus which patient did not relapse, and we could look into the tumor cells in the patient which are relapsed versus non-relapsed. So we could look into how these tumor cells which are present in relapse, oncofetal high relapse patient versus low, what are the difference in them? So as you can see, some of the interesting features are tumor cells in oncofetal high have cell mobility, which could be EMT marker, embryo developmental like genes, so could be more stem-like cells, and again, EMT as such, while the areas in non-oncofetal are more metabolic active. So now we could go back to our stereomix map, and where we have already plotted oncofetal score from high to low. We could also plot now expression of this relapse associated cell signature, and we could find that a lot of these relapse associated cells were present in the same neighborhood as oncofetal ecosystem is. So suggesting that there's a reciprocal signaling happening between a tumor cell which first created oncofetal niche and then it bring back to this EMT-like reprogramming in the tumor. And we could validate some of these things by using single molecule FISH. And again, I'm not going into too much detail, but now we are using this technology to look into the not only the early stage tumor where we know that after surgery some patients relapse and not relapse, but we are recently funded by NHMRC to do this in a big cohort of immunotherapy patient where we are looking at tissue-based biomarker of patient where this new immunotherapy EXO plus Pava may or may not work. So as we know, it works only 30% of the patient, but we do not know which 30% patient this immunotherapy is working. And this is where we are biopsying every patient. We're going to do spatial transcriptomics to understand if the presence of these oncofetal cells could predict where immunotherapy will work or not. Just to summarize, in today's talk, I presented that oncofetal ecosystem is associated with immunosuppressive microenvironment. And now we are showing that this high oncofetal score is associated with therapy response. And we could identify these VEGF-alpha and PD-L1 positive cells in oncofetal ecozones, which may serve as a biomarker of therapy response in SCC. So this is the work done mostly in my lab in Perth and with strong collaboration with Floron in Singapore and Z in Shanghai. And we are strongly working with Liver Cancer Collaborative in Western Australia. And special thank to BGI team, Lonky, UKIN, and BC for collaborating on this stereomix work. Thanks a lot.
Wonderful.